Assalamu alaikum. I am Dr. Sabikun. I have said Professor Bashundhara Ad-din Medical College. Today I am going to discuss about the protein metabolism. It is a very important topic. So, protein metabolism depends on basically two important concepts, that is protein turnover and amino acid pool. Protein turnover, the rate at which body proteins are constantly being degraded and again resynthesized. The normal protein turnover is 300 to 400 gram per day, that is 1 to 2% of our body weight. 75% of the liberated amino acids produced by degradation of body proteins are reutilized. That means, whenever the protein degraded, 75% of which are reutilized and 20% catabolized, that replaced by body protein. So, what is the definition of amino acid pool? Amino acid released by hydrolysis of dietary or tissue protein or resynthesized de novo. De novo means newly synthesized, mixed with other free amino acids and distributed throughout the body. That whole things, that whole the points are known as amino acid pool. I am again repeating, that is amino acid released by hydrolysis of dietary or tissue protein or newly synthesized amino acid mixed with other free amino acid and distributed throughout the body. They constitute the amino acid pool, which is about 100 gram. So, what are the sources of amino acid in the amino acid pool? Body protein give actually 400 gram per day, from dietary protein 100 gram per day, and synthesized or newly synthesized amino acid, it varies. The amount of newly synthesized amino acid is actually it varies, and where this amino acid used or utilized in our from the amino acid pool, that is for the newly synthesized body protein, synthesis of porphyrins, creatine, neurotransmitter, nucleotides, etc., and others are catabolized to form glucose, glycogen, carbon dioxide, urea, and ketone bodies, fatty acid, and steroids. So, if I categorize them as anabolic or catabolic fate, the anabolic fate of amino acids are synthesis of non-essential amino acid, synthesis of tissue proteins such as structural proteins, enzymes, hormones, synthesis of non-protein nitrogenous substances that are purine, pyrimidine, creatine, and porphyrin, synthesis of urea from ammonia via urea cycle, synthesis of glucose, glycogen via pyruvate and TCA intermediates, TCA means tricarboxylic acid cycle, and synthesis of fatty acid steroids via acetyl-CoA. And there are also some catabolic fate of amino acid. So, catabolic fate are transamination, deamination, and removal of amino group as ammonia, catabolism of amino acid carbon skeleton, that is pyruvate and TCA cycle intermediate, for the synthesis of pyruvate and TCA cycle intermediates if glucogenic amino acid, acetyl-CoA and acetoacetyl-CoA if ketogenic amino acid, and carbon dioxide, water, ATP after oxidation in TCA cycle. So, transamination, the first catabolic fate of amino acid, that is known as transamination. The definition of transamination is the transfer of amino group from an amino acid to a corresponding keto acid with the simultaneous formation of a new amino acid and a keto acid. That means, there is amino group will transfer from a amino acid to a keto acid, and the amino acid that gives the amino group will become the keto acid, and the keto acid, another keto acid will take the amino group to form a new amino acid. A very common example is the conversion of alanine to pyruvic acid and as well as glutamate to alpha ketoglutarate. In this reaction, serum glutamate pyruvate transaminase, or another name is alanine transaminase enzyme is used, and here coenzyme is the pyridoxal phosphate. So, features of transamination: catalyzed by a group of enzymes known as aminotransferase. So, transamination, the group of enzymes that catalyze the transamination reaction, usually known as aminotransferase. These enzymes are from the enzyme group transferase enzyme, and here the coenzyme used is the pyridoxal phosphate, that is a vitamin B6, active form of vitamin B6, and this transamination reaction also known as both catabolic and also anabolic. Catabolic because here essential amino acid convert into pyruvic acid, and anabolic because here non-essential amino acid synthesized. So, this is a amphibolic cycle. That means this cycle have both catabolic and anabolic potential. Compartment where the transamination reaction occurs is the cytoplasm, and site is the liver, kidney, heart, skeletal muscle, brain. So, if I asked anyone that is where this non-essential amino acid synthesis, the answer will be name the organs where the non-essential amino acid synthesis, the answer will be liver, kidney, heart, skeletal muscle and brain. So, mostly 3 keto acid participate in this transamination reaction. These keto acids are alpha ketoglutarate, oxaloacetate and pyruvate, and alpha ketoglutarate is the keto acid of glutamate, oxaloacetate is the keto acid of aspartate and pyruvate is the keto acid of alanine. All amino acid undergo transamination reaction except 3 amino acids. These are lysine, threonine and proline. These 3 amino acids never take part in transamination reaction. So, importances of transamination reaction is very much important for both clinically and also the some basic importance of biochemically. So, first importance is the funneling of amino group of different amino acid to form glutamate. So, at first the target is to funneling of amino group, the target is to synthesize glutamate. Why we have to target to synthesize? We have the target to synthesize glutamate because it is very easy to break down of glutamate, as because glutamate dehydrogenase is an enzyme is available in all the cells. For this, the catabolism of glutamate is the most easiest way. For so for this we have the target to synthesize glutamate, and formation of carbon skeleton, that is keto acid, that later on catabolized and oxidized into generate energy. That means we also can get energy from the amino acid, and synthesis of non-essential amino acid. So, as we know that essential amino acid are essential because they can't synthesize inside our body. So, essential amino acids must be supplied through diet, but non-essential can be synthesized inside our body. So, in which metabolic cycle this non-essential and amino acid synthesized? So, the one cycle that can synthesize non-essential amino acid is the transamination reaction, and this is the way of integration of metabolism. This is the way of integration of metabolism among carbohydrate, protein, fat, since keto acid can form compound common to their metabolic cycle. So, the basic importances of the main importance of transamination reaction is the for synthesis of glutamate and formation of carbon skeleton that catabolized to generate energy, and synthesis of non-essential amino acid, and it is the integrated metabolic pathway. And clinical significance of transaminase. There is the very two important enzyme that used in the transamination reaction is the alanine aminotransferase and aspartate aminotransferase. These two enzymes also as regarded as a liver function test to see the hepatocellular function. If any damage occur in the hepatocyte, if any disease due to any disease cause, then these enzymes are found in the plasma. Okay. So, serum aminotransferase such as serum glutamate oxaloacetate aminotransferase and aspartate aminotransferase or serum glutamate pyruvate aminotransferase, also called alanine aminotransferase, has been used as clinical marker of tissue damage with increase in serum level indicating the increased extent of damage. That is all for today. Thank you.